For very many years, scientists could alter the mouse genome any way they wanted using mouse embryonic stem cells, ES cells, and gene targeting technology. Now, CRISPR genome editing enables scientists to alter the genome of virtually any organism, and with much greater efficiency than was possible using ES cells. Remember that in bacteria, it's possible to generate an amorphic allele of any gene in the genome as long as you know some base pair sequence at the 5' end and the 3' end of the gene. Using molecular biology techniques, DNA sequences at the 5' and 3' ends of any gene can be attached to either end of a gene that encodes a drug resistance protein, like the ampicillin resistance gene. When these linear DNA molecules are mixed with bacteria, some of the bacteria will take up a DNA molecule, and in some of those bacteria, recombination enzymes will line up that DNA molecule with the identical endogenous gene sequences in the bacterial chromosome to perform recombination. A homologous recombination event at each end of the linear molecule will result in replacement of the endogenous gene with the incoming DNA, here resulting in a gene which is mostly deleted and replaced by an ampicillin resistance gene. Rare bacterial cells in which the recombination has occurred can be selected for by plating the bacteria that were exposed to the linear DNAs on medium containing ampicillin. The linear DNAs have no origin of replication, so the only way that ampicillin-resistant bacteria can be recovered is if the linears are incorporated into the bacterial chromosome. A PCR assay can be used to determine if in a drug-resistant bacterial colony the ampicillin resistance marker was incorporated through homologous recombination. This technique is called gene targeting. Scientists wanting to make mutations in mice adapted this technology for use in multicellular organisms. The key to using gene targeting in mice was to have cells that could grow in a culture dish and then be used to make mice after their genomes were altered. Embryonic stem cells can do just that. To generate an amorphic allele of any mouse gene, gene targeting can be performed in ES cells. Because ES cells are diploid, after homologous recombination, the cells are heterozygous for an amorphic allele and a normal allele. These ES cells can be injected into early-stage embryos called blastocysts, and then the blastocysts are introduced into a surrogate mother. When the pups are born, you can see that they are chimeric, meaning that their bodies are made of cells from two different sources, ES cells from the Petri plate and cells from the blastocyst. And these cells have different genotypes. The ES cells are heterozygous for an amorphic or knockout allele and a normal allele, and the host blastocyst cells are homozygous for normal alleles of the gene in question. If some of the germline cells of the chimeric mouse originate from the ES cells, then some of their gametes will contain the amorphic mutant allele. Progeny that come from such a gamete will not be chimeric. All of their cells will be heterozygous for a normal allele and an amorphic allele of the gene. Homozygous knockout mice can be obtained by crossing two such heterozygotes. Recall that gene targeting can also be used to alter the genome in other ways. For example, a point mutation can be introduced into a gene using a gene targeting construct that contains the mutation. The selectable marker is cloned within an intron and flanked by LOXP sites. After gene targeting in ES cells, the enzyme CRE is expressed from a transgene already in the ES cells. CRE performs site-specific recombination between the LOXP sites, thereby removing the marker gene and leaving only a short LOXP site in the intron that will not interfere with splicing of the primary transcript. The end result is a copy of the target gene that has a point mutation, a so-called knock-in mutant. Scientists have used many different creative variations of the basic knockout and knock-in techniques to manipulate the mouse genome. However, because ES cells that grow robustly in the lab are available only for mice, these manipulations have largely been limited to mice. Researchers have recently figured out how to exploit a naturally occurring bacterial immune system called CRISPR to make any kind of genome alteration in virtually any organism. The CRISPR system has two components: an investigator-designed single-stranded RNA called the single guide or sgRNA, and a protein called Cas9. At the 5' end of the sgRNA is a 20-nucleotide sequence that is complementary to a target site of interest in the genome to be altered. The 3' end of the sgRNA binds specifically to the Cas9 protein. The Cas9 protein and sgRNA are introduced into cells. They get into the nucleus, and through complementary base pairing, the sgRNA brings Cas9 protein to the genomic DNA target. Cas9 makes a double-strand break in the target DNA. Repair of the break by non-homologous end joining often results in a small addition or deletion of a few base pairs at the break. Such a mutation can cause a loss of gene function, a knockout of the gene. Making knockouts this way is incredibly efficient because it doesn't rely on homologous recombination. CRISPR can be used to make knock-ins also. If linear DNAs corresponding in sequence to the DNA flanking the break are introduced into cells at the same time as the Cas9 sgRNA, double-strand break repair by homologous recombination can incorporate that DNA into the genome at the break site, generating a knock-in. Making knock-ins this way is much more efficient than gene targeting in ES cells because the double-strand breaks that Cas9 makes promote recombination. Because no ES cells are needed, and because the genome editing is so efficient, the CRISPR system can be used in all kinds of different organisms. The sgRNA in Cas9 can be introduced into cells or organisms in a number of different ways. For example, scientists can inject genes for sgRNA and Cas9 protein, or even preformed sgRNA Cas9 protein complexes into newly fertilized eggs that subsequently grow into whole organisms with modified genomes. Scientists have already shown that the genomes of human embryos can be modified efficiently using CRISPR technology. One day soon it might be possible to use CRISPR as prenatal gene therapy to correct mutations that would otherwise result in disease.